Vincent, can you tell how many people are using the YouTube video?
I can, yes.
Okay, just wondering.
Yeah. So Susan Willett is here. Welcome. All right, so welcome to the 796th monthly meeting for EPAASM. Of course, this is our Norman Willett Memorial Lecture organized by the Education Committee. And today we have Dr. Carol Bascom-Slack who will give us a talk. And before we go into introduction, I just want to remind you that because ASM is providing a lot of resources, I just want to encourage you to join the ASM membership. It will advance your science career and nurture this network and community. And as you have experienced, we also have the ASM Connect that allows you to connect with your peers and participate in discussion and also members-only events. And we do have the ASM Awards and Prize for 2023, and the nomination deadline is June 20th. So it's coming up. So if you have someone that you want to nominate, go ahead and go to asm.org/awards. And of course, we would love for you to join the EPAASM branch. Last year was our 100th year, and so we can continue to provide this program because of the members, the student chapter, the education committee, as well as the executive committee. Of course, we have the monthly meetings. We have our annual Philadelphia Infection and Immunity Forum, our clinical symposium, as well as the scientific outreach and education program. And as a matter of fact, we've done this poll for the last few meetings. I just want to launch this again to kind of gauge the participants, your membership status, as well as a couple new questions at the end, because Jason and Kyle are working on the next year program, as well as seeing if we can get the in-person meeting going again. And so please answer those questions, and so we can gauge your interests. As a reminder, you can ask your questions toward the end, and you can either go to the participants tab and then click on this little blue hand, and then we will unmute you so you can ask your questions. You can turn your video. Alternatively, you can type your questions in the chat, and we'll relay that, moderate that to the speaker. And from that, I just want to say welcome Susan Willett, who is here participating with us. And I will turn it over to Bobby McHale, who is the co-chair of our education committee.
Thank you, Vincent. My name is Bobby McHale. As Vincent said, I am the co-chair of the education committee. Vincent just showed you some of the breakdown of our branch, and we are a very active branch. There's no question about it. My co-chair is Cynthia Keeler of Delaware Valley University. Unfortunately, she could not join us today, but just want to give you a little bit of background why we have the Norman Willett Memorial Lecture. It's a long-standing tradition of the branch that each year the education committee would sponsor a education-focused program that would appeal to educators both at the high school level as well as up through postgraduate education. The concept was originally suggested many years ago by Dr. Norman Willett, who was the co-chair of the education committee at that time. Dr. Willett was a professor emeritus of Temple University Dental School. He was president of the branch from 1975 to 1977, and a strong advocate for involving students in science education from kindergarten through high school. To this end, Dr. Willett developed science modules that students and teachers could take back to their schools. In order to facilitate these programs, Dr. Willett was the recipient of many grants, including Howard Hughes grants and NIH grants. Dr. Willett passed away in 2014, but his legacy lives on through these annual presentations in his honor. So as I said, each year the Education Committee invites a renowned speaker in the field of microbiology education to share his or her views with the branch and the guests. This afternoon, we're delighted to have Dr. Carol Bascom-Slack as our speaker, and one of our committee members, Rebecca Sheridan, has had the opportunity to work with Dr. Bascom-Slack, and she'll provide the introduction.
So thank you for being here. Dr. Bascom-Slack and I actually worked together on her research-based program called PAIR, which is the prevalence of antibiotic resistance in the environment. And this is a course-based research program that was meant to engage both undergraduates and high school students together so they could get an authentic classroom or real research experience like real scientists would have as undergraduates and high school students. And I had the luxury of having a microbiology lab and 4 microbiology classes. So for 3 years, I participated in the PAIR project. And I have to say that my students definitely benefited from being able to do this authentic research and contribute to this large database that is both international and in the United States that Dr. Bascom-Slack has developed. In fact, I was just saying a few minutes ago, it also meant that my AP Stat teacher was able to use our data and have the AP Stat students also involved in, doing the statistical analysis of a real research project, not just one that they make up for them to have data. So her research program has definitely touched many students across the country. And I personally am really thankful that I had the chance to be involved in this. And so I would like to, formally let you go on and talk about your crowdsourcing environmental surveillance of antimicrobial resistance and your course-based research program. And thank you for allowing my class to be part of this for those 3 years.
Thank you, Becky. So Vincent, shall I go ahead and start?
Yes, can you?
Shall I?
Yeah, go ahead.
Yeah.
Sure.
So, okay, can you see my slides okay?
Yeah, that's good.
Great. All right. Well, thank you very much for the invitation. I'm honored to be here. And thank you, Becky, for that awesome introduction. I'm thrilled to hear that some of the data is still getting analyzed by stat students. So I'm going to get right into it and just start with a little case study that some of you may have heard of, may be familiar with. This case occurred several years ago where a 62-year-old healthy woman was admitted to the hospital after 9 days with stomach pain and pretty severe diarrhea. And it was a pretty easy diagnosis. The doctors determined that she had food poisoning from salmonella. She was given Cipro, which at the time, this was 1998, that was a big gun. Salmonella hadn't developed resistance to Cipro at that point. And so they fully expected that she would improve, but 5 days later, she got worse and not better. And turns out the bacteria had caused a perforation in her intestine, and some of the bacteria from the gut leaked out into her bloodstream. So they had to do surgery to stitch up the intestinal wall and gave her 2 additional antibiotics in addition to the Cipro. And the salmonella was cleared, but the patient ended up dying from sepsis. A culture after autopsy revealed that the patient was in fact infected with multi-drug resistant salmonella. It was a strain called DT104, and this was the first of several infections that occurred in Denmark in a few weeks, led to an outbreak of, I believe it was 27 patients in total, and a couple of them died. So this story, unfortunately, isn't a unique story. We're all familiar with antibiotic-resistant infections and the toll that they take. But what was special about this case was that it was the first case that I'm aware of where a pretty definitive link between antibiotic use on farms and a antibiotic-resistant infection in humans were linked. So it was a pretty nice, tight connection. And that was made possible because this occurred in Denmark. So Denmark, just about 1 year prior, had instigated a very, very robust surveillance system that many of you may be familiar with. And so it's pretty much farm to table surveillance. There's, I think, almost every flock of chickens and pigs are tested for salmonella. Every patient with a salmonella infection gets tested and so on and so forth. And so in about mid-June, there was an alert that a slaughterhouse had come up with a salmonella that was multi-drug resistant. It was this DT104 strain that had been floating around the EU, but hadn't really reached Denmark. And again, that was why they had implemented this surveillance system. It was to try to keep this strain at bay. And it was right around the same time that the outbreak was starting to blossom in the hospital. There were, I think at that point, 5 patients with this DT104 strain, usually resistant to 5 different antibiotics, but this particular variant was resistant to 7 different antibiotics, including Cipro. And so some nice detective work ensued, and DNA sequence analysis revealed a point mutation that was the same in all of these strains. It was an aspartate to asparagine in the gyre gyrase gene, which is of course the target for ciprofloxacin binding. So this case study is really helpful in the classroom to illustrate the concept of One Health, which kind of forms the context for the PAIR program that I'm going to talk about. One Health is really the concept, it's been around for a long time, that the environment and plants that we eat, the animals that we eat, and human health are all inextricably linked. And as you're aware, there's a large percent of antibiotics produced in this country go to livestock operations. The FDA has really reined in use for growth permission, but there's still a lot used for prophylactic and treatment of sick animals. And the CDC estimates that 1 in 5 resistant infections are caused by bacteria from food animals. So there's large presence of antibiotics. I don't need to tell this crowd about natural selection. So there's this selective pressure to select for resistant microbes. The antibiotics get excreted from the animals that we eat. They get into wastewater and whatnot. We know that farm workers from Stuart Levy's seminal work, and I think it was 1976, farm workers that work on farms where antibiotics are used harbor higher levels of resistant microbes in their gut and on their skin than others. So we can kind of see that there's a problem, but what can we do about it? There is action being taken, but I felt like as an educator, I wanted to do something more. And I can tell you a little bit about my my trajectory in a moment, but first I'll tell you that from an educational point of view, I believe antimicrobial resistance provides a really great context in which to teach evolution and natural selection, as I just mentioned. It's not the normal textbook example, but it provides a really great example that is relatable to students. So the PAIR program was developed really in response to some of my concerns about this public health problem, if you will, and also based on my prior experiences. So the program really combines some educational goals that I'll discuss a little bit more, but primarily the goal is to make research experiences more equitable and accessible, and some scientific research goals, quite simply put, at its most basic, to identify resistant hotspots. What are the areas in the environment that we should be worried about? And then flag those areas for stewardship activities. And this then provides additional backdrop for education research projects that I have ongoing. And I should probably say that the education research that I do is really my bread and butter. That's where I get my funding. That's primarily how I spend my time. So I can talk a little bit about that at the end if there's time, but I'm guessing I won't have time to talk about that. But the work with PAIR has really been the catalyst for a lot of the education research questions that we ask. So just a little bit about undergraduate research. again, I probably don't need to tell this crowd, but there's many 20+ years of evidence linking all sorts of positive student outcomes with a research experience. Persistence, retention in STEM, self-efficacy, feelings of belonging, being like a scientist. However, there are some issues with this, and that is that students usually seek out an undergraduate research experience late in the pipeline. They do so after they've decided they want to be a STEM major. I know I, for one, was a junior when I decided, basically based on conversations I heard with my peers, that it's about time I get a research experience. I didn't really know what that meant, but I knew by junior year, it was something I should probably do. And I embarked on this process by knocking randomly on the doors of different professors who I had had as teachers in classes I took. In addition, many students are excluded from that type of research experience that I just described, this apprentice style research experience, for many reasons. First is just a lack of understanding that this is something you should do. As I mentioned, I figured this out only because I kind of heard conversations from my peers, and I started understanding just naturally that this was something I should be doing. But if we were in person, I would ask all of you if you had an undergraduate research experience. And those of you who did, I would pry a little bit and ask how you landed that research experience. And what I've heard are often answers like, well, my parents were scientists and they told me I should do this, or my parents had a friend. I knew I was interested in STEM, and my parents had a friend who worked at a university and I got to work in their lab, and so on and so forth. So this is a form of social capital. You have those connections that many students don't have. So there's a level of exclusion. Many students need to be earning money in their out of class time. They cannot afford to do an apprenticeship in somebody else's lab unless they're going to get paid for it. As a result of understanding this more clearly, I now don't have undergraduates work in my lab unless I can pay them, because it's just not fair. And then there's just not enough research labs at many universities for the number of students who are interested in a research experience. So, for example, at big state schools, there's often far more STEM majors or interested students than there are research slots available. And at community colleges, their mission is to teach, not really to do research. And so often there isn't great research infrastructure in community colleges. And given the fact that depending on where you get your data, about half of the undergraduates in the US go through the community college system. If those students are not offered a research experience, then again, that's not fair, that's not equitable. So enter course-based research experiences, in which the research experience is embedded into class time, into the course. And this is thought to make research more equitable, because you don't need to know that this is something that you should do. It happens naturally in the classes that you take, and you don't need to be losing income by getting your research experience in a course. And it, of course, solves the problem of not having enough research labs if it can happen in a course setting. However, there are some issues with course-based research, and I worry that it may not be equitable for the reasons that I'll describe later. But let me just ask a few questions. Let me see if I can launch my poll. Goodness. I can only see. Okay. So I'm going to launch this poll, and I think you can probably see all the questions at once. Is that true? Are you seeing the polling questions? Okay, so the first question, how many of you have just heard of course-based undergraduate research experiences? They're often referred to as CURES. And then the second question, is that have you ever used or implemented a cure in a course that you teach? And then, of course, this isn't a very scientific poll. I didn't want to ask a million questions. So I'm not asking how many represent 2-year and 4-year institutions. But if you have implemented a cure, what best describes your institution type? And I'm sorry, Becky, I do not even have high schools on here. If you teach at a high school and you've implemented a cure, maybe answer for 2-year institutions. So it looks like about 2/3 of respondents so far have heard of cures. So for those of you who haven't, I'll describe a little bit more. And the majority of people, about 2/3 of the group here, has not implemented a cure. And it looks like of those who have, no one is representing a 2-year college. Of course, I don't know how many people in the audience are at 2-year colleges, maybe nobody. More people at private institutions than public, but again, we don't know what the total numbers are. So I'm going to end this poll now. So that gives me a sense of, oh, I have to share the results. Sorry, now you can see them. So first of all, what are cures? So I will probably be talking a lot about cures, and you'll get the sense of what they are just by nature of listening to me talk about PAIR. But I will say that everybody's definition of cures is a little bit different. So this is some of the research that we've done investigating faculty barriers to implementing research in their course. It turns out that there are quite a lot of barriers and quite a lot of motivators. Most faculty that have heard about cures think that they're a great thing and they'd like to do them. And yet many faculty have not done them. And those who have tried them often discontinue use. So that's some of the research that I do, is trying to understand the psychology of motivation. Why is it that people that want to do cures don't do them? Why is it that those who try them and discontinue, to help us understand how we can develop better cures that are easier to implement, lower barrier, and so on and so forth. Getting back to the polling results, I don't know if we may just not have good representation from 2-year colleges, but generally, I didn't finish answering the question about what a cure is. So definitions differ, and some people have a very high bar definition of a cure and what it is. And not surprisingly, if your definition is pretty stringent, you're less likely to have tried using a cure. And so I will just say right off the bat, there isn't a standard definition. Those people who research cures are trying to come up with one. I personally think of a cure primarily as an experience where the outcome is completely unknown to the instructor and to the student, and the outcome should have real-world relevance, meaning stakeholders outside the classroom should be interested in the results that the students are getting in their classroom experience. But that's just my personal definition. So getting back to the poll results, it turns out that generally cure use is more prevalent at baccalaureate institutions, private institutions, less so than at 4-year public institutions, and less so than at community colleges, probably for some of the reasons I mentioned earlier. And given the statistics that I just told you about the number of undergraduates who go through a community college, we are not really providing these undergraduates with a research experience, even though the idea, the notion of infusing research into coursework was thought to be a more inclusive opportunity for students to do research. So yes, it's more inclusive, but it's not inclusive enough. So I wrote my questions out here just in case the poll doesn't work. So there's this premise that with cures is to make research more equitable. But if certain institutional demographics preclude cure use, then we might be further widening disparities. And so this is kind of backdrop for why I've developed PAIR. But let me just give a little bit more background on course-based undergraduate research experiences, or CURES. So they tend to fall in two categories: those that an instructor creates from scratch, and they're implemented generally in their own class, often based on the instructor's own research. And you can imagine it's a fair amount of work to develop this. They can be costly, and some of them are not costly. There's also what have been referred to as network cures. These are developed by an individual or a team and then disseminated for use by others, so that the methods have kind of been worked out, the protocols optimization has been done, and yet the students are not doing the exact same thing. Sometimes the word parallel research projects is used, in that students are using similar or identical techniques, but each student is doing something a little bit different. So that would be under the category of network cures, and pair is under that umbrella. Here are a whole bunch of other network cures. You might have heard of Tiny Earth, Small World Initiative, CPHASES, Genomics Education Partnership. So there are many of these, most of them in the life sciences, but few in chemistry, and probably a couple in physics. So I'll just tell you a little bit about my personal trajectory so you understand the background that I'm coming from. So I started teaching, gosh, about 20 years ago. I was teaching introductory biology at Yale. So this is the course that all of the pre-med students take. It had at that time about 300 or 400 students in it. And I was teaching an upper level microbiology course. And I don't know, about 5 years into teaching, there was a report that came out that said that just under half of the undergraduates in the US who entered college intending to major in STEM actually majored in STEM. So we were losing about half of those interested undergraduates. And I thought to myself, maybe I'm part of the problem. I'm taking the first biology class that these students take. I'm teaching very traditional lecture style. It might be pretty boring, actually, to be sitting in the audience. You know, this was 20 years ago, so the rise of Google and the internet. And as faculty, we no longer needed to be the sage on the stage delivering all this information. And it was kind of the birth of more active lecturing styles. So that got me to thinking about my own personal teaching. And then a few years later, so I started becoming more interactive, implementing active learning in the classroom. But then a few years after that, I started working with a colleague at Yale who had received an HHMI professorship to run a really cool research-based class that was called Rainforest Expedition and Laboratory, or REAL, as we called it. And it was amazing. There were about 15 students who got to travel to South America, all expenses paid. We sampled plants. We isolated endophytic fungi that were growing within the tissues of the plants, developed assays to try to isolate active compounds. Antibiotics being some of the compounds we went after. So 15 students, 4 faculty members. So I call this a very boutique course-based research experience. This was absolutely not scalable and absolutely not disseminatable. So that then got me to thinking about issues. It was a great experience, but it got me thinking about issues of scalability and sustainability. What happens when the funding goes away? And right around that time, Jo Handelsman started working at Yale, and I went and knocked on her door and I told her what I was thinking. And she said, perfect timing, because I have this idea for a course. And remember how I mentioned earlier that usually students get a research experience a little bit too late, after they've already declared a major. So there was this growing notion that we want them to have research experiences in the first 2 years, those critical years, so that we prevent the attrition that we see. And so Jo's idea was to create a course-based research experience for non-majors in the first year. And so I was lucky enough to get on board with that. And that launched at Yale in 2012, and it was called Microbes to Molecules. And then it was later disseminated and was kind of renamed Small World Initiative. And then it's subsequently been renamed again, and it's now called Tiny Earth. So many of you may have heard of that. And it was basically crowdsourcing to discover new antibiotics. And it's still running today. It's very successful. And that was a great experience. But I went to conferences a few times and talked about this program. And I would bump into instructors who would say, I would love to do Small World Initiative or Tiny Earth, but I don't have enough control over my course to change it around. It was a full semester course. Or instructors would say, I don't have the bandwidth to just reinvent my entire course. So I started thinking about how to decrease faculty barriers even further. So having a disseminatable network here was thought to reduce faculty barriers, but that wasn't enough, clearly. So that was when I started thinking about something very small, a chunk that would be meaningful. So I spent a lot of time thinking about a research project that could be meaningful and generate meaningful data, but wouldn't take a lot of time. And so that's when I came up with the Prevalence of Antibiotic Resistance in the Environment project, or the PAIR project. And it now consists of several different, like a library or suite of modules that are all short duration, in which an instructor can kind of pick and choose what they want to do and how they want to do it. But it started with what we refer to as the core module. And in this module, students culture bacteria from soil samples in their geographic location and subject those samples to serial dilution, plate on medium containing antibiotics, do some simple calculations to determine the number of colony forming units that grow on the antibiotic containing plates over those that grow on non-antibiotic containing plates, to get the percent in their sample that are resistant. And they then upload that data into our database, and it displays in live time onto a map, which you can see here. Here's some of our soil sample data. Each dot represents a student entry. It's very interactive. And so to date, we have about 160 undergraduate institutions and thousands of undergraduates annually that are entering data into the database. And participation ranges from community colleges to R1s, private, public institutions. And we now have about 10 international partners. And the idea was to just blanket and get really broad geographic coverage. There's just not a lot of systematic environmental surveillance happening. So there's great clinical reporting, the World Health Organization, CDC, so on and so forth, but not a lot of environmental sampling. And yet you read papers and reviews, and there's always a call for more and better and robust environmental sampling. So I talked to a lot of people. They said, well, we don't do it because we don't know how, we don't know what culture, what type of medium to use. And I thought, it's a student research project, let's just try something. So the project was born, not perfect, but we've been learning, and it's been a lot of fun, and we're collecting a lot of data. So there's really two overarching research questions. We want to look for correlations. We want to find hotspots. We want to ask whether individuals living in areas with high levels of resistance are at greater risk of developing a resistant infection, and whether we can identify areas for which stewardship efforts should be invoked. And of course, there are surveillance studies that are done, but they're usually confined within a small geographic location. We're trying to answer big questions with a pretty shallow data set. These are student researchers. But we hope that over time, we'll accrue enough data that we can actually answer some big questions. And we're just in our early stages of data analysis right now as we speak. One of the big problems with this has been that all of the existing data out there by different research groups, they've used different methods. And so you can't compare the results from one research group to another. And so our students are all using systematic methods so that we can compare across all of our data points. So the methods for the core module were thought out such that they would be simple, relatively low cost, not time consuming, and importantly, they use techniques that are normally taught in a microbiology classroom. So instructors don't have to do, a lot of times there's a certain mindset of techniques or skills or concepts that instructors want to teach, and they don't want to vary from that. And so this uses concepts and skills that are pretty routine in a microbiology lab. So students, as I mentioned, they collect soil from a geographic location in their vicinity. They subject the soil to serial dilution because we don't know how many microbes are in each soil sample. They plate onto plates without antibiotic and with. We're using tetracycline, 2 different concentrations, 3 micrograms per mil and 30. And then they count colonies on the countable plates, do their calculations, as I mentioned, and upload into the database. So, and Vincent, you'll tell me if there are pressing questions about if there's any confusion. Okay, because I may not monitor the chat. So this is just a representative sample from Reza Farrow's group at Bellevue College, Community College in Washington State. And the students are plating onto McConkey plates here. And you can see these plates we would consider probably the most countable. They're going to do their colony counting and calculations and enter into the database. And then in real time, there's a curation process that occurs. We require, for example, each soil site to be sterol diluted once, but plated twice. So there's a replicate. So we require, before the data will display, it has to meet certain criteria. There has to be a replicate. Those 2 replicates have to be within a certain range of each other. They can't be wildly different, and so on and so forth. And so the darker dots represent a higher percent resistance. So you kind of see the map here. I should point out that we've changed our data upload process as of about January, February of 2020, which was terrible timing because we're no longer displaying the old data. I need to transport the old data into the new database. So the new database is great because it's easier to use and it displays in real time, but we launched it right when COVID struck. So a lot of our faculty members had just started PAIR and then they couldn't execute. So it was really almost a full year of not collecting data. So I'm kind of proud that we have the amount of dots that we do because we lost a bunch, or didn't actually lose any data, but people were just unable to produce data. I picked this example for you because this is from Newman University in Pennsylvania, and it shows you if you click on these dots, it pops up with the name of the students and the actual, so in this case, 7.8% of the bacteria that grew were resistant to 30 micrograms per mil of tetracycline, and so on and so forth. There's more data screens behind this one. So that just gives you a little sense of the data that the students are collecting. And I'll say that from the beginning, my goal in this project was, as I've kind of alluded to, is to target instructors, to make a program that was low barrier for instructors to begin implementing course-based research. We didn't expect to have great student outcomes in such a short duration program, but we're pleased to see that we do, and I'll talk about that a little bit. But what we've subsequently done is create a whole suite of short duration modules like the one I just described. Some of them, the ones in yellow, are what we call dry lab. They are virtual or computer-based or pen and pencil and paper based. So they were really good during COVID. These 2 are molecular based. This is also molecular based, but it doesn't generate authentic data. These are BSL-2. These are occur subsequently on tetracycline resistant isolates. So we have a discussion with instructors who want to do those to make sure that they're up to safety standards. So I'm looking at the time. I'm just going to whiz through and talk a little bit about a couple of the molecular modules. So I've just told you about the culture-based detection of tetracycline resistant colonies. We've also worked in collaboration with the company called Mini-PCR to develop some molecular-based modules for detection of tetracycline resistance genes. And this has been great because it has allowed us to include a lot more high school groups. So Becky Sheridan, who introduced me, she ran it in a high school and it was amazing. And an experience like most high school students don't get. Most of our high school instructors do not have the appropriate biosafety measures in place to be able to work with isolation of environmental unknowns that are resistant to tetracycline or other drugs. So in the molecular sampling, students isolate total metagenomic DNA from soil, and then with that metagenomic DNA, they can run a PCR reaction on it. So there's no culturing whatsoever. And they're just looking for presence or absence of tetracycline resistance genes. So there's also a case study approach that does not produce authentic data, but it primes students for the activity that I just described, the metagenomic DNA PCR study. And so in the case study known outcome module, it's under the auspices of a narrative that's based on an amalgamation of 4 or 5 different actual research papers, one of them being the one that I mentioned at the onset. The idea is that there's been a multi-state outbreak of carbapenem, in this case, carbapenem-resistant E. coli, and officials trace the source to some tainted pork, and neighboring farmers want to act proactively to determine if their soil contains the resistance genes. And so students go through this narrative. They're given, I'll just kind of crash through this. They're given tubes with quote metagenomic DNA isolated from these farms, but it's actually plasmid. They're given primers against the plasmid sequence, and they're looking to determine which farm, if any, has the gene of interest. And the backdrop for this is the NDM1 gene that was first isolated in New Delhi. It also makes another case study that we talk about. You can see that just in less than 10 years, this is showing the prevalence just in India in 2006, 2008, and then how it spread worldwide. So this is another, we have a teaching video on this that kind of helps students understand the spread of antibiotic resistance genes. So I'll just quickly go through this. As I said, the students are given tubes with what they're told is metagenomic DNA, but it's actually plasmid. And then they run 2 controls and the 2 farm samples. That prepares them for the authentic sampling approach, where they're looking for 2 different tetracycline resistance genes. So in this research module, students take their soil sample, and it can be the same one that they used in the culture-based techniques in the culture-based module, if they choose to do that. Isolate metagenomic DNA, use primers directed against 2 different TET genes, TET-A or TET-M. So one is an efflux pump, the other one has ribosome binding, so 2 different mechanisms of action that they can talk about and discuss. And again, the workflow I just described, and that data is uploadable into our database as well. So we're creating data on tetracycline resistance gene presence or absence. Now we're working on doing the same thing for other clinically more relevant genes, including NDM1. And we're doing that in collaboration with James Kirby at Harvard Medical School, who is an infectious disease specialist. So quotes from students doing various modules make me think like a scientist, and it opened up for more options for me in the future. If dental school fails, I could join a PhD program. We contributed to a real life science experiment, and it made us feel important. It's from a high school student, and so on and so forth. So students tend to give favorable responses. And this is really old data, but just showing, this is from high school students compared to before doing the pair research. How likely are you to take another course in science, apply to a college or university, become a scientist? So the blues are positive and the reds are negative. We have qualitative data that shows that the project is career or major affirming. So it doesn't always change minds, but it affirms their decision to be a STEM major. for example. And this is from undergraduate students. So again, keep in mind, this question was asked about the core module, which is a 2 to 3 class period experience. So we're not talking about long duration. And as I said, the target here was instructors, not students, but by developing over time, the suite of different modules, the idea was instructors could start with the core module, get that under their belt, and then in subsequent semesters, they could add on other modules. And it's pretty flexible. So we have data that shows that the vast majority of instructors who begin with the core module go on to expand, and they use multiple modules. And in fact, many of them are now running full semester cures. They started with the single core pair module, and now they're doing full semesters. So that's exactly what we wanted. So we're pleased about that. I'm just going to put up a little bit of quick learning gains data. So again, keep in mind, the duration of this program is very short, but the students do show significant learning gains after completion of the core pair module. They're small, but they're significant. And this is probably a little small to see, but we have a completely virtual module called PairSeek that was created almost 100% by a talented undergraduate named Scarlett Bliss. And she did analyses and showed that she saw similar learning gains after completion of just this module, no other modules, similar to what we saw for the core module. And she asked a lot of demographic questions and did some really nice regression analyses. And she showed that learning gains were not really driven by any demographic factors. But she also asked a STEM identity question. This was developed in a published paper. It's a single question where you are asked pre-participation and post how you identify what your identity is. So this is a STEM professional, and this is me over as a 1. And this is a overlapping Venn diagram of the STEM professional and me. And so she sees similar gains in that, not particularly driven by any factors, except, interestingly, students who are working, especially those who are working more than 10 or 20 hours a week, show lower gains in STEM identity. So this is a problem that we've started thinking a lot more about, how external work is affecting students' learning and their STEM identity and whatnot. So I've told you about the institution type participation. So this is just a pie graph showing that distribution, and some quotes from instructors. One of our favorite quotes talks about how this was the catalyst. Another one talks about it being the activation energy they needed to get over the hump to implementing course-based research. But we do have our challenges. There is quality control of the data issues that I could talk for another hour about. But we have really implemented a bunch of curation steps so that we can throw out data that we don't feel are robust. And we do tell everyone that we consider this a first pass. If we see any interesting results, obviously we'll need to go in and investigate them further. Data management itself is a big task. And as I mentioned, this is not my primary, this is not what I'm funded to do. So it takes a lot of bandwidth. And another challenge is just staying innovative, because I am primarily an education researcher now and trained as a microbiologist. And so I need to really collaborate with other microbiologists to stay relevant and make sure that what we're doing is what's needed and what's appropriate. So I welcome any feedback from anybody in the audience, or contact me later if you have ideas for things that we should or shouldn't be doing, because that's really what drives us. And then just quickly, future directions. I mentioned one of them, water sampling. We've got a group working to streamline protocols for water sampling, the identification of clinically relevant genes listed here, MCR1, some of the beta-lactamase genes, metagenomic analysis of nanopore sequence data. So I'm collaborating with Amy Pickering in the Department of Civil Engineering at Berkeley who uses nanopore sequencing of environmental samples to look for all of the antimicrobial resistance genes present. So we're trying to develop a module that would be scalable and economically feasible for our students to do, that would allow them to do their own sequencing. And I've got an undergraduate now, Leah Rihari, who's working on a data analysis workflow. So we've got all of this 3D geospatial tag data, and we'd like to overlay it and compare to all of the many other existing geotag data sets out there to look for some correlations. And so she's working on that now. So that's it. Thank you very much. And I'm happy to take any questions.
Thank you, Carol. That was a very interesting talk. So let me ask a question first. So I really love the program and also your questions to begin with, right? So so how do you link or imagine looking at how the antibiotic resistance you find in the soil or the water linked to the infection rate or more downstream of stewardship? I was wondering if you could like maybe partner with any CDC database or like working with epidemiologists and looking at that kind of infection data.
We would love to work with epidemiologists. So send them my way. Yeah, so those are great questions. I think that the first research question that you mentioned, which was the first on my slide, connecting our data to health outcomes, is a pretty big lift. Let's be real. But I think that's the overarching goal. And I think it's possible when we accrue enough data points to use some of the existing databases out there. There are national databases, but there are also local ones. So for example, Tufts University has just combined forces with WellForce, which collects data from lots and lots of hospitals in the area. But the problem with that is we don't have enough parent institutions in the Boston area. But I can envision we have some geographic regions where our presence is really strong. So maybe getting a hold of some more localized data sets would be a start. But that's what we're kind of in the process of exploring now, is how can we best analyze this data and make some correlations. In terms of stewardship activities, I think that's maybe a little bit of a lower bar in that we want to try to identify hotspots. The clinicians that we've worked with have indicated if we can go after some of these rare, kind of traditionally clinically rare antibiotic resistance genes, and if we see spots in the environment where they're pretty prevalent, then that could be a red flag, and it might be a great opportunity for intervention. But that's probably going to require more than just me, right? I'm going to need to get policy people involved. Love to get the ear of the USDA. So this is where I'm kind of dependent and reliant on making connections.
I think Amy, did I unmute you?
There we go. Carol, that was really lovely talk, and congratulations on giving so many students a great opportunity to dip their toe into the research waters. I've just returned from the ASM microbe meeting in Washington, D.C. And so I'm you know how you are after you come back from these meetings, you're very inspired. So what you're talking about, this connection between agriculture and the clinic and animal health, was captured by a theme called One Health many, many years ago. And One Health is enjoying a little renaissance right now. So it could be useful for you to Google people who are involved in One Health. And I'll bet some of them are epidemiologists. And I know we have ASM members who work at the USDA. We used to have, the Eastern PA branch used to have a meeting every year that was a government person. And I thought, was it Olare who used to have a USDA connection? I can't remember. But anyway, I think it's worth kind of noodling around and see. And if I can look at my notes from the meeting and remember anything, I'll reach out to you. But I think there are people interested. I also think that if you know someone who's writing a big NSF grant, connecting with your CURE project could enhance their greater impacts, because that's
Right.
usually
Yeah,
educational.
that's a great idea. We do have, the reason this is not, the program isn't funded is, as you know, we got funding to start the program.
Right.
But I made the mistake. So usually the progression is you write to build something
Right.
in the education world, and then you write for a next phase of funding to disseminate it and grow it. But my idea was that it could be disseminatable very easily. And so when it came time to write for that second grant, I thought, what do I ask for? And it's obviously the data, the science part of it, the data collection is not robust enough to apply for an NIH or a sciencey grant.
Right.
So it's a great idea to kind of tag along with somebody who's doing research, more traditional hard research, as their greater impacts section.
The other thing I would suggest is to reach out to the editor-in-chief of JIMBY, Stanley Malloy. He's at San Diego State, and see if there's different types of articles for that journal. And so even if yours is a little bit more informational and less data, it could be a way to create a community that would be in a conversation with you about this. So I would take advantage of those resources the ASM offers already in terms of networking. But that would, I would suggest that, because what you're doing is really great.
Thank you.
So I could plug one more time the ASM Connect. So maybe you can use that channel to connect with other people. All right, so I have a few comments and questions. So Dieter said checking hospital bed linen might be more fun if you want to find a lot of antibiotic resistance, might even have a greater effect on influencing students' life choices.
Sorry, say that again. I didn't hear that.
Checking hospital bed linen might be more fun if you want to find a lot of antibiotic resistance.
Right. Yeah.
And then Vita's, okay.
No, I was just going to say, my original idea was to do something on the human body or to go in hospitals, and then quickly decided it's going to be a little tough. But yeah, I agree with the
And Vita asked, in this study, do you rule out the animal sources in the soil of TET resistance bacteria?
Do we rule out animal sources? I think that it's the opposite. We're expecting that a lot of them will be animal sources. And in fact, I didn't get into this, but we do use McConkey media. We used to use nutrient broth augers. So we're going to be selecting or enriching for coliforms. And so we do see that soil that has been trod by animals harbors greater CFU counts and greater resistance levels, if that answers the question.
Yep. And then Joanne said, what is the best way to get involved as a high school teacher? She's beginning a micro course spring 2023. I'm former molecular biologist. We have a PCR machine and definitely equipped to jump into what you've mentioned here. I have a grant source that I can request funds for. Should I write, I think it's a contact for CTSC at Tufts. So
yes, that you can just Google pair Tufts, which is my institution, and you'll find my page, and there's a contact form there. Or if my email address is in the announcement, I can put it in the chat. You can email me directly, and yeah, we can have a conversation and get you set up. We can have a conversation about where would be the best place for you to start in terms of which module to start with. But yeah, absolutely.
Great. And then two more comments. One from Earth saying, great talk and a fitting subject to honor Norm's legacy. And on the same idea, Susan Willett said, my dad would have loved this. His interest in microbiology began when he learned about microbes in the soil. He grew up on a farm. And as others have mentioned, he loved bringing science, microbiology in particular, into schools. And she recently found some pictures of him during one of his teacher sessions, and he looked so happy. And he was always interested in targeting folks who are underserved in science education. So thank you.
Nice.
So with that, thank you very much, Carol, for giving a wonderful talk, even all the way from Greece. And if Bobby would like to say anything, we can.
Yes, thank you, Vincent. And Carol, thank you very much, especially your long distance conversation. Fortunately, the technology held out and it came across very well. And
Great.
I think you've given us some ideas, certainly some of ideas for some of the education committee outreach that we do. So thank you very much. And Vincent,
Great. And
provide her email address. That would
Yeah.
be really helpful.
yeah, I can put it in the chat right now.
Okay.
Yeah, well, thank you so much. And again, I welcome any feedback. Send me questions or comments by email. That's great.
Thank you.
Yeah, thank you very much. And
Thank
thank you
you.
all the participants for another year. And we'll see you September.
Thank you.